One.
Hello everyone. Welcome to this webinar from the School of Biological Sciences at the University of Southampton. So let me start with some introductions. I'm Lindy Holdendye. I'm Professor of Neuroscience and I'm head of the School of Biological Sciences. And just to give you a little bit of background, our school covers quite a wide range of biological sciences research. So everything from biomedical sciences and dementia and cancer biology and microbiology to developmental biology, molecular and cellular biosciences, all the way through to ecology and evolutionary biology and biodiversity. So there's a lot of different research activities going on in the school. And this provides a really exciting environment for our students to receive their education for their degree programs, because our ethos is very much about the research influencing the education that we provide for our students. So you are connected to research that's going on in the school. And so, you know, as part of this, what we're doing with this webinar is we're providing you an opportunity to hear a little bit more about one aspect of the research in the school. And that is from Mark Chapman, who's with us this afternoon. Mark is an associate professor in the school, an associate professor in the ecology and evolutionary biology group. And he says on his webpage that he's working out what genes do. But he's actually working out what genes do in very important context in terms of how we can optimize crops. And as you can imagine, you know, feeding the world in the 21st century is one of the major challenges that we face. So optimizing crops is a really important area of research. And so he's going to talk to you about his research now. He's going to provide a research presentation for you, and you'll have lots of opportunity to post questions as well. So do post your questions, and if there's a question that you haven't posted but you see one that someone else has posted in the chat and you really like it, then like it with a heart, and then I'll be able to pick up on the most important questions or the most liked questions for Mark to address in that order. So we're hoping this is going to be a really interactive session, as far as it can be, of course, on the internet. So do participate and post your questions. And I think with that, I don't think I've forgotten anything. I'm ready to hand over to Mark.
Thanks very much, Lindsay. That's great. So really interesting, really exciting to be here. So the plan today is I'm going to tell you a bit about the type of research I'm doing. So what we're going to talk about is the theme of domestication, so how we get the species that we eat today, where these came from, how we know this, and then what this actually means for our food security going forward. As Lindy said, you can post a question at any point. I'll probably wait until the end to answer all the questions. And just a quick reminder, don't post things about your individual submissions or admissions. If you save those for the admissions tutor, that would be best. I certainly can't answer any questions like that in any detail, and you probably don't want to advertise those questions today. So if you post a question, it will come up on my screen and we'll get to those at the end. So I'm just going to go ahead. I'm going to share my screen in a second. So just telling you a bit more broadly about what I do. So Lindy said that I work out what genes do. And what that means is our genomes have got, a human has got 35,000 genes, and we probably know what about a few thousand of those genes do. And it's even less for other organisms. So we really need to know what all these genes are doing, how they're interacting with each other, and how different species are interacting with each other at the same point. So what I'm trying to do is actually take an interesting question, whether it's about evolution, whether it's about ecology, or whether it's about crops and the foods we eat. And we're trying to narrow down and say, right, this is a gene that's really important. It might be a gene that controls protein content in a legume. It might be to do with drought tolerance, or it might be a gene in the wild that has given rise to a new flower color or a new type of plant or something like that. So I'm going to go ahead, share my screen now. I'm going to share my slides, and hopefully if it doesn't show up, someone will let me know that it's not showing up. Right, so I'm going to go ahead now and start talking about, this is partly my research and partly my field of research, so not specifically things I've done. And what I'm going to talk about is what we know about domestication, so where our food comes from, and how we can actually use that to plan how we might combat potential food insecurity in the future. So I'm just going to talk about what is domestication, and you've probably all got an idea of what domestication might be, but I'm going to use this definition. It's the process of adapting wild plants and animals for human use. So pretty much anything in a supermarket or in any old market that you're going to go to, this is things that we eat, and they've had to come from somewhere. They've evolved from wild species. And we're not just talking about food. We've got species that we use for fibers like cotton, and I can imagine that probably half of you are either in your pajamas or you're in your jeans, and they're probably made of cotton. So species like that, we have to remember it's not just about food. So why have we domesticated species in the first place, and what have we domesticated, and what particular traits are of interest to us? So these are just some examples. So we've got lots of food crops, anything you find in a supermarket. We've got fibrous crops. It might be flax, it might be hemp, it might be cotton, things like that. We've got lots of species that we take advantage of because of the colours they produce, and we can use them as dyes. And when we think about animals, we've got animals that pull carts, or there may be even things like greyhounds that we enjoy watching in a race or something like that. So we've got all these species we've been domesticating, and for all of these different types of reasons. But there's other reasons as well. So 10,000 years ago, when domestication of a large number of crops took place, it wasn't as if some people sat down and said, I want to make that species look better, taste better, grow more fruits, be taller. It was just something that took place over time. And humans were selecting for particular individuals that maybe ended up having slightly bigger fruit. Maybe they were more resistant to a pest, or they just fitted into the local environment better. Maybe they grew really well on a rubbish heap outside, something like that. So there's all these other traits that kind of got tagged along. So we think about domestication being for its final use, but we've got all these other traits that go along with it. So harvesting, crops that are harvested, or wild species that are harvested more easily, will be the ones that humans decided to select and ended up keeping and growing. It might be behavior. So certain wild animals are potentially much more hazardous than others. I don't think anyone's tried to domesticate a tiger, for example. That's possibly a bit too risky. There's things like storage qualities. So any crop that's going to break down and rot in a very short time might not be ideal. And there's actually some wild species that are potentially mildly toxic, and humans have domesticated these species which have lost their toxicity at the same time. So this is just some examples. So on the left here, we've got some examples of very large sort of normal salad tomatoes and very small wild tomatoes. So the wild tomato has a fruit that's about the size of it's not much bigger than a pea, about maybe the diameter of a 20p coin, something like that. And you can imagine that maybe 6,000 to 10,000 years ago in the Andes, where this domestication happened, humans were selecting the larger fruit because it's much more economical to collect half a dozen large fruit than it is to collect 50 small fruit. At the top example here, this is a safflower. It's got important seed oils, and the wild relative is very, very spiny. And you can imagine that humans, if they came across a plant that was less spiny, they would have potentially collected the seed from that plant, not all the spiny ones around it. At the bottom here, we've got wheat, and you can see that wild wheat has a very tiny ear on it, and the domesticated wheat has a much larger ear. So over time, we've selected for larger and larger wheat grains on the awn of the plant. And on the right, this is two examples of types of rice. And there's something called shattering. So most wild species need to distribute their seeds. So they will naturally drop their seeds when their seeds are ripe. But when humans were about the time they were domesticating species, if they had to scrabble around on their hands and knees to pick up every last seed, this was very inefficient. So if a mutation arose that prevented these crops or these wild species at the time, from dropping all their seeds, so a non-shattering variety, these would have been the ones that were collected by the humans. So over time, we've got things like larger fruit, less spines, bigger edible parts, and a loss of shattering. And this is quite common in a lot of the crops that we've domesticated. And these are some of the other examples. So obviously selecting for individuals of animals that have more docile personalities, and selecting beans and other things which are less and less toxic. So over time, we've got food, we've got companions, we've got all these dyes and things like that. But there's lots of different traits that humans have selected as we've gone on this journey. So what I'd quite like to do is just show you some examples of wild species. And what I was hoping to do was maybe just have you think about what these wild species might have evolved into. So top left, we've got a little fruit. It's only about 2 centimeters in diameter. It's yellow. Actually, lots of people look at this and say it looks a bit like a tomato. Yes, it does, but it's not a wild tomato. It's a wild aubergine. So if you've been to the supermarket, the average aubergine is probably maybe 6 to 8 inches long, big, purple. The wild species has tiny fruit. They're quite bitter. They're quite tough as well. Top right, so actually wild species related to this do live in places in Britain, and this is actually a wild lettuce. So this is the progenitor of the lettuces that we eat today. So quite a significant morphological change over just a few thousand years of evolution. Bottom right, so obviously you could probably guess that that's a wild sheep, and therefore we've got our domesticated sheep there. They're not a million miles apart, but you can see that the domesticated sheep doesn't have any horns, so it's potentially less harmful to the farmer. This in the middle, you may have seen along roadside verges in Britain. That's actually the wild relative of cabbage. So a massive morphological change, going from a weedy plant with very little edible parts to this cabbage where you get obviously the big head on the cabbage that we would buy in the supermarket. Bottom left, we've got beans, and I've just shown you a wild bean, which is very tiny, and a domesticated bean. And then what I hoped I would do was actually ask you what this final species is. So if you can see my mouse, there's a species here. I was hoping you'd put that in the chat, but because I'm sharing my screen, I now can't see the chat for some reason. So what I'm going to do is just give you 5 seconds to think about it. So if you're in a room with your friends or your relatives, what do you think this species on the left has been domesticated into? Lindy, maybe if you see anyone commenting, you want to shout?
I'm looking for comments, but I can't see any, and I don't know what it is, Mark. I'm afraid.
Well, some people can't.
So I can't help.
It's not an asparagus, but some people do say that.
Yeah.
It's a corn. And actually, these images are to scale. So the corn on the left, this is a wild species called Teosinte. It's got very small, edible, but very, very hard seeds. And on the right, obviously, this is an ear of corn like you would buy in a supermarket. So over just a few thousand years, we've got this massive morphological transition. And because of the amount of money that maize is worth, there's actually been an incredible amount of research into this crop. So this is just a nice example of the massive changes we can have during domestication. So this is, I just mentioned cabbage. Its Latin name is Brassica oleracea. So on the left, you might have seen this along motorway verges, and it's been domesticated into all of these species. So all these 6, plus the wild species on the left, they're all one species. So we've got kohlrabi, where humans have selected for sort of a fat, quite tasty, quite crunchy stem. We've got kale, where humans have selected on the leaves. Broccoli, cabbage, broccoli and cauliflower, where humans have selected on kind of modifications of the buds and the flowers. Brussels sprouts, they're actually the buds that are growing on the side of the plant. And then cabbages I've mentioned as well. So all of these, one species, maybe only 4,000 years of evolution and these incredible morphological changes. Can anyone think of another example of where we've got one wild species and an incredibly diverse array of domesticated species? Potentially an animal. Anyone got any ideas? I'll go straight into telling you the answer anyway. Dogs. So dogs all came from one wild progenitor, a wolf, and over a few thousand years, potentially 12 to 15,000 years, we've got tiny little toy dogs, we've got big dogs that go running up mountains with barrels of brandy around their neck, and everything in between. So again, incredible morphological diversity over just a few thousand years of evolution. So where did it occur? So one of the most well-known regions of the world is called the Fertile Crescent. And you can see why it's called the Fertile Crescent, because it's a sort of C shape that goes from Iran and Iraq through the Syrian desert into the north of the Nile. And actually, one of the reasons that domestication probably took place here is because of the local environment. So there's these very big rivers, there's the Tigris and the Euphrates that go through Iran and Iraq, and the Nile in the west, obviously going down towards through northern Africa in Egypt. And this is probably where the first domestication of species took place. And actually dozens of species were domesticated in this region. So some of the big hitters like wheat and barley and chickpeas and peas, and also animals like goats and pigs and cows, they were most likely domesticated all in this relatively small region of the world. And there's dozens of actually more minor crops as well. So lentils and carrots, we've got dates, apples, lettuces. So all of these were most likely domesticated in this one very small part of the world. But it's not just that one part of the world. So everything in orange here is one of the centres of food production. And the Fertile Crescent is indicated there. But all of the other orange areas are where we think there's been large numbers of crops domesticated. So we've got parts of China around the Yangtze River. There's a potential domestication centre in New Guinea, large parts of Africa. We've got the northern part of South America, and we've got Mesoamerica and North America. And these are just some of the crops. So we've worked out pretty much where most crops have originated. And this is largely down to knowing where their wild progenitors live. And I'll tell you about that in a second. But why was it in these places? So I already mentioned that the Fertile Crescent has some very big rivers. It obviously has a warm but not too hot climate and so on. But also, we can't domesticate every species. It's not possible. So there's some level of luck as to the right species being in the right part of the world where the soils and the climate are good as well. So just a couple of examples. So horses have been obviously domesticated, but zebras haven't. And you might look at those and say, well, why is one domesticated and one not? Well, it turns out zebras are particularly vicious. And my interesting fact for you is that zebras injure more zookeepers every year than tigers do. And part of this reason that they can't be domesticated is that behavior. But also their eyes are just slightly further back on their heads, not by a lot. And you look at a zebra and a horse and you maybe wouldn't notice, but they've got slightly better peripheral vision. And you can imagine that a few thousand years ago, to actually domesticate one of these species, someone would have had to have thrown a rope around its neck and potentially tie it to a tree, at least. That might have been the very early stages. And it would be basically impossible to do that with a zebra, but relatively easy to do that with a wild horse. So two species, at face value, they're very similar, but actually one is probably undomesticatable and the other one is obviously horses, and they have been. Another example is almonds and oaks. Again, they're not a million miles apart in terms of the fruit or the edible part, but the wild species of both are poisonous. But one is poisonous because it's got one gene that controls poison, and one is poisonous because it has dozens of genes that control the poison. So if we need a mutation to occur in a gene to knock out poison, it can happen very easily in an almond because you've got one gene, and if the important mutation comes along, you knock out the poison. But to do that in oaks, you'd have to knock out multiple genes. So we've domesticated almonds because of this, but it would be very difficult to domesticate an oak. And there's other things in animals too. So animals with a very long generation time, very specific diet, or very poor disposition in terms of what humans are looking for, we would generally avoid those species. And when we talk about diet, you think of something like a panda. It's got such a precise diet, basically only eating bamboo, that it would be inefficient and possibly impossible to domesticate a panda. Sounds nice to have a panda in your home instead of a dog, but probably not a wise choice. So as I said, partly it comes down to rivers and soils and climate, but partly it comes down to the disposition and the physical and the genetic characters of the species that exist there at the time. And another interesting fact is that 88% of humans, they actually speak languages that are related to those found in only 2 of those domestication centres, the Fertile Crescent and the Chinese origin of domestication around the Yangtze River. So 88% of humans across the world potentially have originated from parts of the world where agriculture began. So how do we know where species were domesticated? I've just given you a laundry list of everything and where it was domesticated, but how do we know? And basically, we presume that most crops were domesticated where their wild ancestors live. So if we can understand what the wild ancestor was, we can then look for its distribution, and we can be fairly sure this is where domestication took place. So in the left picture here, I've got wild soybean shown in red dots. So we're fairly sure that this is where the domestication of soybean took place. And on the right, all of the dots show where different wild tomatoes live. And we're pretty sure that the tomato was domesticated somewhere along the western coast of South America. Now, both of these are a bit imprecise. So now we've got genetic tools, we can look at the genetic similarities between crops and whole big numbers of wild populations of the wild ancestors. And we can try and pinpoint exactly where in the world it took place. What I'm going to do is tell you a bit more about animal domestication. So again, in a similar way, we can use genetics to say this is the closest relative of these domesticated animals. Where does that wild species live? So here's just some examples. This is pigs, sheep and llamas. So pigs, we've got domestication in the Fertile Crescent and potentially a second domestication in China. But they're within the distribution of the wild pig, which is this massive grey area going from Spain and North Africa to China. So we don't know exactly where domestication happened, but we can try and guess that. Sheep, they're also from the Fertile Crescent, and again the genetics says that it overlaps with the wild species in the Fertile Crescent. And then llamas, we've got the west coast of South America too, so actually quite similar to where tomatoes are domesticated. But if we use genetics, we can more pinpoint exactly where this event took place. So this is what we call a phylogenetic tree, and this is looking at the genetic relationships between lots of different types of maize or sweet corn and its wild relatives too. So we've got this big group at the top, and basically if things are very close on this diagram, they're genetically closely related. And as you get further and further apart, they're genetically more distantly related. So all of the maize forms one group. So we've got a domestication of maize that gave rise to all the maize we have now. And then we can try and say, well, what's the closest wild relative? And I've shown here there's a couple of populations of maize, wild maize, that come from a place called Oaxaca, which is in Mexico. And actually, if we look at where this population grows, it's from a very, very small place in Mexico. So because of the genetics, we can say, instead of it coming from somewhere in Mexico or South America, we can actually pinpoint a very small place called the Balsas Valley, which is in Mexico. So we've managed to pinpoint exactly where domestication potentially took place. So how did it take place? Well, as I alluded to before, it wasn't a conscious decision. There was no goal. So humans couldn't have looked at a wild species 10,000 years ago and said, I want that to look like X, because there was no X, nothing had been domesticated. So it was more about collecting a few plants, growing them around a camp, maybe even some of them were dropped as seed, they just fell off the plants that were being collected. And they started to grow up in close proximity to where the humans had set up their camps. It was occurring at a time when the climate was unpredictable, so it might have been actually necessary for humans to have a more reliable source of food. And also about this time, humans had exhausted a lot of the big game. So a lot of the big animals that would have been full of nutrients may have actually been lost, or at least if they're not extinct, at least a lot of them had been removed, for example, a lot of them being eaten almost to extinction. So there were all these things kind of happened at about the same point. And in the Fertile Crescent, we're talking about 8 to 10,000 years ago. But we think it took place quite gradually. So it wasn't a case that suddenly there was a barley that fitted the bill. It had massive seeds, it was pest resistant, didn't fall over in the rain, anything like that. It's very unlikely that it could and would have happened like that. And instead, it's more likely to be a gradual process. And something we can do is we can look at fossilized remains of wild species and crops from the last 10,000 years, and we can look at how traits have changed over time. So this is just some examples. It's quite a complicated graph, so don't worry too much about all the details. But basically, we've got time on the bottom axis. So we're going from a long time ago on the left to recent on the right. And we've got two different sets of traits. So you remember I said about non-shattering, so species that don't drop their seeds. So over time, clearly we've got an increase from no non-shattering, i.e. they had the wild phenotype, they dropped all their seeds. And over 4,000 or 5,000 years, we've got an increase in the amount of non-shattering, such that the most recent collections from archaeological remains are clearly from non-shattering plants. But this clearly took a couple of thousand years. And then we've got grain size as well. That's a really important trait for any domesticated species. And we've got here again the same species. We've got barley, 2 wheats and a rice. And in all of the examples, over a couple of thousand years, we've got an increase in the grain size. So it didn't happen overnight. It didn't happen very, very quickly. It took a couple of thousand years to go from essentially a wild trait, so dropping its seeds and having small grains, up to a domesticated trait, essentially not dropping its seeds and having much larger grains. So what happened in these parts of the world, and in particular the Fertile Crescent? So actually, because agriculture had started, humans had more time to do other things. They weren't spending their whole time hunting game, gathering grains or anything like that. They had a more predictable source of food. So they had some farmers, but then other people could establish other trades. And in the Fertile Crescent, this is where metal tools have first been recorded, where we think writing's first recorded. So you can imagine the hieroglyphs in Egypt, for example. This is part of the Fertile Crescent. And then we started to get empires being built up. Villages turned into towns, turned into empires, essentially. And then with this extra time and this extra technology, armies started to be developed. And then some of these domestication centres, this is why they've taken over large parts of the world, because they were at an advantage in terms of their technology, in terms of their numbers, and in terms of the tools and things like that. So they could actually invade and take over neighbouring hunter-gatherers, potentially to get better land, different types of crops, things like that. So what do we know about genetics? So this is kind of where my research comes in. So there's an established sort of series of events that takes place during domestication. So wild species generally have very large distributions, so they have lots and lots of genes and lots of different variants of that gene. So you can imagine in humans, we've got lots and lots of different heights, tall to short. We've got different hair colours, different eye colours. So lots and lots of variation. But if we were going to select a few humans to start the next generation, and I'm not saying this is a good idea, but we would only have a subset of the genetic variation. So we might pick, tall people with blue eyes or shorter people with brown hair. And over time, if we keep doing this, we lose all the hair colour, the eye colour variation, and we end up with a population which has got less variance. Now, when we domesticate things, we do this semi-deliberately because we don't want to hang on to the traits for shattering. We don't want to hang on to small seeds. We want our crop to actually be to fit into our lifestyle better. But by doing this, we lose a certain amount of genetic variation. So this is just an example. This is measuring genetic variation. Don't worry about what the exact values mean. But this is some work I've done, and we did some work in sunflowers. And we've basically got a lot of genetic variation in the wild sunflowers. When we've got these primitive lines, so these are grown by Native Americans, they have a bit less variation. And then when we look at the improved lines, so the high oilseed lines, we get even less genetic variation. So over time, we've got this drop in genetic variation. And we can use this to actually find genes that look like they're important to the domestication process. And this is something I've been doing for probably a decade now. And these are some examples. This is not my particular research, but these are some of the really important findings we've got. So there's a gene called teosinte branched 1 that's been found. So teosinte, as I said, is the wild progenitor of maize. So this is just a diagram of what teosinte and maize look like. And there's this gene which prevents branching in the domesticated maize. But when they sequenced this gene, there was no genetic variation. And they thought, well, how do you have a gene with two identical sequences, but it controls a very different plant morphology? And it's basically due to the gene being turned up. So if you ramp up gene expression of the TB1 gene, you lose all the branches. So very simple genetic change. We've gone from a very bushy plant to a very sort of single stemmed plant, which ends up having much bigger ears of corn on it because of that. Here's another example. So I'm going to talk about maize because it's just super well studied. This is a very small but simple domesticated maize on the left, and this is the wild progenitor on the right. And you'll see it's brown on the right. So these aren't exactly equivalent because this brown thing is called a glume, and it covers over the edible part. And actually, to get the edible part out, you have to kind of smash it with a rock to really get the edible part out. But over time, we've gone from this very inefficient, rather potentially quite starchy sort of thing on the right to this sweet and potentially super tasty thing on the left. And one of the genes involved in that breaks down this thing called a glume. So there's a gene, they've called it teosinte glume architecture just because it's a gene that affects the glume. It doesn't change the morphology of the whole ear, but it loses that glume part. And if you lose the glume part, it's much easier for a human to harvest and enjoy and eat it. And if you look at a piece of corn, so if you've ever had corn on the barbecue or just boil it or steam it, you know when you get those bits stuck in your teeth, that's the glume. So in a domesticated corn, we have small and papery glumes, but in a wild maize or a wild corn, they're very, very hard, and you need to basically smash them open with a rock to get anything out of the corn. And again, that's a single gene, and this one does have a mutation in it, and it basically just changes the sequence of that gene, and you get big hard glumes versus small papery glumes. This is an example from dogs. So we've said in maize, there's two genes. One is ramped up in expression to change the branching, and one of them has a mutation that changes the sequence. Here's an example from dogs where it's neither of those. We've actually got more copies of a gene. So in our human genomes, we also have some variations that aren't just changes in the base pairs. They are changes in the number of the genes we have. There's a gene called amylase. It breaks down starches. And in wolves, they only have one copy of this gene. And they've looked in 35 wolves, and they all only had one copy of the gene. If you look in dogs, you get a variation between somewhere about 5 up to about 30 copies of this gene. So many, many, many copies of this gene. So what's this likely to do? Why do they have more copies? Well, if you have more copies, the gene expression of that gene is going to go up. So we've got two graphs here. One shows that the expression of that gene goes up. So dogs have more of that gene being turned on. And on the right, we've got the actual activity of the enzyme. And again, in dogs, the activity is much higher. And we presume this is because when dogs were domesticated, they were possibly fed on scraps, which might have been more vegetable based, whereas a wild wolf has got a largely carnivorous diet, and it's very unlikely to be spending its time eating corn waste or vegetable waste or anything. So humans, by selecting for dogs that are potentially better adapted to a human environment, they would have selected for dogs that had more copies of this gene too. I just wanted to wrap up by saying, what's the future of our food? So we, in a period of climate change, the temperatures going up, we're potentially going to have more drought. So we need to think about this in terms of breeding crops that are climate resilient. One thing we can do is simply breeding crops. So crossing together the individuals that we're interested in and isolating offspring that have certain combinations of traits. Another possibility is genetic modification. Whether or not you think that's a good idea or not, it is definitely a potential way to make a stress tolerant crop. But there's something just recently talked about called neo domestication. So you take a wild species which is tolerant to something you're interested in, maybe a drought tolerant, maybe a pest tolerant plant, and then you either insert in the genes that you want, or you can breed in the genes you want. So if we know the genes that make bigger tomatoes, bigger ears of corn, tastier food, we can actually find those genes, put them into a wild relative that is already tolerant of some environmental situation like a drought or something. So there's been some examples, and I'm just going to tell you about a stress tolerant tomato. So tomatoes aren't actually particularly stress tolerant. They don't handle droughts and very high temperatures very well. And in this example, they took a wild tomato, which was already fairly drought tolerant, and they put in, just using genetic modification, so it's more of a proof of concept. You're not going to be finding this in your ketchup, but they put in the genes that increased fruit size, that increased the amount of fruit, and they increased the amount of lycopene, which is quite an important compound. It's found in tomatoes, but they managed to ramp it up. So by doing this, they managed to triple the fruit size. They managed to get 10 times more fruit from the plant, and they got a 5-fold increase in this lycopene content as well. So it's just two pictures here. On the left, we've got the wild tomato, and then the tomatoes with the fruit size genes introduced. And on the right, we've got a picture of the flower head, and you can see the wild species on the left in panel B has quite a small number of flowers on a stem. But on the right, if you introduce the right genes, you get a very large number of these flowers. So we've got a lot of potential. I'm not going to say that we have to GM everything. Personally, I have positive views of GM, but I think there are lots of options that we can start to investigate. So I'm going to finish there. I've gone for 35 minutes, so that's enough for most people. I will just end by saying this. So if you did have questions about admissions, you can just talk to Amrit directly. Amrit's our admissions lead. So if you have any questions now, you're welcome to ask me or Lindsay. But if it's specific to an admissions question, please direct that straight to Amrit, and she's the ideal person to ask. So I guess, do I go back to Lindsay now? Is that right?
Yeah, thanks, Mark. That was terrific. So we're able to take some questions. I can't see any questions in the chat at the moment. I've got a bit of an echo on the line.
I'll stop presenting this. I might be the one who's echoing anyway.
Yeah, if you just, yeah, okay, that's good. Thank you. Yeah, oh yeah, we got a question. Hang on. Let me put it in the chat. So the question is, Daisy asks, what's the difference between GM and Neo Domestication? Thanks for your question, Daisy.
So GM to me is taking a crop and putting a gene in, and that gene could come from anywhere. So it could be making a tomato, going back to tomatoes, that is resistant to a certain pest, and that gene could come from anywhere. It could be a bacterial gene, it could come from a different plant, it doesn't have to be related to tomatoes, it could come from a bean or a maize or something. Neo-domestication is more about identifying the genes in a species that are required to make a domesticated version of that species and putting them into a wild species. So there is genetic modification, there is the potential for genetic modification to do that, but you could do that through breeding. So you could cross a wild tomato that's pest resistant to a domesticated tomato. And over time, you would increase fruit size, fruit number, and everything like that. But GM is typically, to me, bunging in a gene from wherever you find it, and it could be anywhere. And again, I say bunging in like I'm, saying it's terrible. I'm not saying it's terrible, but that's sort of how I imagine GM to be.
Yeah, so it could be from another species, for example. It could be anything. Yeah,
exactly.
Yeah. Thank you, Mark. While we're waiting for some more questions to appear in the chat, just give people to have a chance to think. Could I ask, what does the neo-domesticated tomato taste like? Does it taste any good?
I don't know. I don't know whether they've actually given them to taste panels. I mean, ultimately, if this was something that was going to be used, it would have to go through the human trials, potentially, feeding it to a mouse first and then to a human. I am not aware, and if they did taste them, they haven't told anyone what it tastes like. So I'd be curious.
Yeah, and so you show two striking indications of same species but looking incredibly different, so dogs and brassica. Can you just comment on how that comes about? How do you end up, in terms of genes and gene expression, how do you end up with same species looking so different?
So we've done quite a lot of work in brassicas, and other people have worked in various species. What it seems to be is that the genetic changes to go from some of those different morphologies are very, very simple, maybe just changing 1 or 2 genes. So the fact that they're really different, so if you take the dog example, it might be just 1 or 2 genes that control coat color, 3 or 4 genes for stature. There might be a couple of other genes that do different things in terms of the tail sticking up versus the hand hanging down. But humans, when they see something they want, they're very good at picking it. So Darwin, when he was writing his book on the origin of species, he had a whole chapter where we talked about domestication, and he talked about pigeons. And he was a pigeon fancier, and he knew lots of pigeon fanciers. And the diversity in domesticated pigeons is crazy. You've got loads of different colors, different feathers, everything like that. But if a human sees something they want, they will collect it and they will breed it. And that doesn't usually happen in the wild. You don't have tiny populations being bred amongst themselves like that. It's quite specific to humans and the traits that they're picking up.
Thanks, Mark. I've got a couple more questions. Do you think there will be a point when genetic modification is necessary to keep up with the rapidly changing climate?
Yes. I mean, so humans, since the Industrial Revolution, 1700s, when climate has started to change, humans have been able to breed just through crossing the relevant strains. They've managed to breed crops that do tolerate different, a gradually changing environment. The problem is that our gradually changing environment is not gradually changing anymore. It's rapidly changing. And there's actually evidence that for some crops, and in particular, I think wheat is one of the examples, that it's starting to lose yield in particularly hot or dry years. 20 or 30 or 40 years ago, there was enough breeding going on that we could keep up with that. And now we seem to not be able to keep up with that, at least in some crops in some parts of the world. So either we need to be quicker and better at breeding, which there are ways to speed up breeding, but it's not as quick as GM, or we need to start taking on things like GM and basically saying, right, we've got a drought tolerant plant and we've got a tomato, and we need to put the drought tolerant gene into the tomato, things like that.
How do you speed up breeding? Because you must be constrained by the life cycle time of the organism that you're working with.
You are, yes, but if you know which part of the genome controls pest resistance or heat resistance, you can instead of growing up thousands of plants to become an adult to then test them, you can grow up thousands of plants and you can quickly look in their genomes and find the ones that have got the right combination.
Okay, so you don't have to wait for them to reproduce.
Exactly. So you might be able to find a plant that has the right bits of the genome for drought tolerance, heat tolerance, large fruits, whatever combination you're looking for. And you can throw away 990 plants and keep 10 that have got the right combination.
Thanks, Mark. What is the outlook for commercial use of GM crops? This person says they saw the Japanese tomatoes recently.
I'm not aware of Japanese tomatoes or whether they are or aren't GM. At the moment, we in the UK we don't grow GM crops. That was part of a European directive, and now we've had Brexit. We actually have the potential to change that, and there are consultations going on at the moment. In the last 5 years, there's a new type of genetic modification come along, and I don't know whether you've heard it. It's called Crispr, CRISPR, and it basically is a way of putting in a gene where you don't have to put in any other bits of DNA. So one of the reasons why the British government decided not that we don't like GM is that a GM crop in the past would quite often have other bits of DNA in it that came from the original donor. And if it's been through a bacterium, for example, there's potentially bits of bacterial DNA in our tomatoes or whatever. The CRISPR approach, you can put in one gene very precisely, or you can knock out one gene very precisely without any extra bits of DNA going in there. So it is almost like speeding up domestication by simply making the right version of a gene that will give us the taller plants, tastier fruits, or drought resistance that we want, without it being this really complicated process of bits of DNA going in there that we can't really keep track of. So my personal view is that it should and it can start to become much more commercially available, but that's something that will take a bit of time, I think.
Yeah. There's another one. I'm trying to keep up with them. If anyone wants to like a particular question, then it will, as I post them, because I'll post them, then it helps me prioritize which ones. But there's an interesting question in there about human evolution, I think, saying that humans must have been very inquisitive to actually go about, bringing plants in and growing them and domesticated crops and animals. Could they also have, acquired traits themselves, I guess, but, through their activity, and, humans will have evolved in the process as well. Yeah,
absolutely. So one interesting thing is that hunter-gatherer populations, obviously, anytime they had a baby, they would have to take that baby with them where they were going. And it'd be quite difficult to carry a baby around whilst trying to hunt down a sheep or something like that. So actually, it's estimated that the birthing window, so the age at which a mother has a second baby, was about 4 years. So at 4 years, the child is old enough to walk by themselves and they're not being carried by the mum. So the birthing interval was about 4 years. With agriculture, it looks like the birthing window went down to about 2 years, because mums could stay at home, or potentially dads as well could stay at home too. Obviously, I don't know what happened 10,000 years ago, but the birthing window got less because humans could have more babies without it being kind of cumbersome to the hunter-gatherer approach. So it looks like humans have adapted, evolved themselves to actually speed up the birthing interval, and this is why some populations 10,000 years ago would have really taken off. But there are other behaviours that we have as well. We're definitely much more aware of things like colours and smells of the food we eat, and I think partly that's come from having a choice, because if you're growing crops and you have enough, and something smells a bit weird and something smells great, you've got the option of eating the great one. But if you're hunter-gathering and all you've got is some tomatoes that are a bit rotten, but you haven't eaten for 2 days, you're going to have to eat those. So I think we've actually adapted to potentially being more sensitive and more choosy in the type of things we choose to eat and drink.
Yeah. So 5 of our audience are interested in this question, Mark. Is it true that insects may have to become a larger part of our future diet? Should we be domesticating insects to make them tastier?
I mean, there's parts of the world that eat insects already, so this isn't something that's completely crazy. Again, I mean, if it's my view, I have no problem with that. They're incredibly nutritious and full of protein, and much easier to rear than the majority of cows and sheep and things like that. So I'm not going to say that it needs to become part of our diet, but I think it would be a very sensible thing to contemplate. Yeah.
Thanks. And will some genes transferred by the Crispr approach be rejected by the plant you transfer it to?
It can happen. So quite often when people are doing these experiments, they will do multiple replicates. So they might introduce a gene into 5 replicates and potentially check, because CRISPR is very, very precise. So it's actually very easy to know exactly where it's inserting. In the more sort of traditional GM approaches, you could insert a gene that interrupts another gene and therefore sort of mess up the plant's host genome. As far as I'm aware, you can quite easily track the changes that you're making during CRISPR. So I think that means that it's unlikely to be rejected unless you planned your experiment very badly and you inserted or modified a gene that turned out to be crucial for that plant.
Thanks, Mark. How can we increase the rate of genetic modification in the lab to produce crops that can keep up with the rate of human population growth?
So that's another thing as well, is that we haven't just got climate change, we've got a growing population. Now, one of the risks associated with that is that cities get bigger and they start encroaching on the good agricultural land. So we've actually got to get more food, but potentially either from less land or from more poor land. And one of the things that is being looked at is things like salt tolerance, because very few of our crops can grow on salty soils near the coast. But if the cities get larger and we need to grow plants on more land, we might have to start using land that is very close to the sea, where it's very high in salt. So I'm not entirely sure what you mean by the increase the rate of genetic modification, but I think we need to know more about species that are already adapted to, for example, salty soils, and that should be a priority. And potentially identifying varieties of crops that are already salt tolerant to a degree. So even if we go in the supermarket and you think, well, every aubergine is within 10% looking exactly the same, if you go to other parts of the world, there'll be long, short, round, purple, yellow, green ones. And some of these will obviously be better or worse in different environments. So there might be genetic variants out there, we just need to find them. But in terms of genetic modification, basically we need to know much more about the genes we're interested in and really find efficient ways to introduce them into the plants.
Do you think it will be more beneficial to focus on adapting plants, livestock to the changing climate, or focus on slowing the progression of climate change? That's a provocative question.
I mean, yeah, nice question. I mean, yes, ideally we need to stop the progression of climate change. I don't think anyone would disagree with that. I don't think anyone is happy with a massively changing climate and just doing lots of genetic modification to keep up. So I think we really should be, and we rightly are, doing a lot to investigate how we can slow the progression of climate change. But we've got all these scenarios and models that are built about what may or may not happen. And even in the best case scenario of almost every country in the world halving its CO2 outputs, we're still going to have a changing climate. And by the end of the end of this century, we might have a 1 or 2 degree increase in temperature at the best case scenario, but worst case scenario it might be 5 degrees. And if we can keep it down to the 1 or 2 degrees, maybe we can keep up with it by doing a lot of breeding, but I think we have to treat crop breeding and GM as something that is a necessity, because we cannot stop climate change even if we do our very best.
Yeah, and it's happening more rapidly than just a typical plant domestication would happen without any help.
And
so
it's going to affect wild species as well. There's going to be wild species that maybe they're at the top of a mountain where climate change is meaning that the region in which they can live is getting smaller and smaller, and you can't migrate from the top of a mountain to anywhere higher because you're already at the top. So climate change is affecting wild species like that too.
Yeah, it's why this topic is just so important, such an important challenge for 21st century. Yeah. Do epigenetic changes play a role in the turning on or off of beneficial genes, or turning on of beneficial genes, or turning off of genes unfavorable to domestication of crops? That's a good question.
Short answer is yes, they can, but I don't think that many of the genes we know about that underlie important domestication traits are epigenetic mutations. And the reason for this is in plants, the epigenetic modifications can be reset. So you could have a plant that looks like it's got a valuable phenotype, it's got larger fruits, but in the next generation, those epigenetic modifications are removed and you go back to having smaller fruits. So yes, epigenetics can change a lot of these traits, but as far as I'm aware, because of their instability, I don't think we rely on any of them for in the traits that the food that we eat.
Thanks, Mark. That was a good question. For CRISPR, is it often only transferring one gene? Or for some traits, such as better heat resilience or drought resilience, would you need to transfer multiple genes?
Yeah, I think the ultimate plan would be to work out a way to not introduce dozens and dozens and dozens of genes. I don't think anyone is aiming to completely change a tomato into a maize or anything silly like that. But potentially, yes, drought tolerance is a very complex phenotype. It's not just about a gene that makes a plant drought tolerant. So it may have to have modifications in its stomata, so it reduces water loss, and it may have to have modifications in its roots, so that it's better adapted at getting deeper down in the soils. So even for something like drought tolerance, possibly picking 3 or 4 genes, maybe 5, 6 genes, and carrying out a genetic modification or a CRISPR experiment in that way would be the best way to do it. Instead of just saying, right, we can only do one at a time, let's go for multiple, I think is the best choice there, yeah.
Yeah. There's an interesting question about transgenics, just posted in the chat. Could you put a gene from a plant, for example, a drought-resistant gene, into an animal to help them cope with environmental stresses like climate change? What do you think, Mark? What about that animal to plant transgenics?
Yeah, I mean, it does follow on a bit from my last question. So it would depend what that gene actually does. So a gene that makes a plant with bigger roots, I don't know what that would do in a sheep, but I can't imagine. It certainly wouldn't initially make that sheep drought tolerant but if it was something about modifying a cell membrane, maybe to make it more or less resistant to osmosis or the transfer of ions from one tissue to another, it could be that that would work. I don't think that anyone knows enough about the genomes of plants and the genomes of animals and how they intersect. I mean, they say that we've got half of our genes are found in a banana, so we do have very similar genes in our genomes. But exactly whether you can modify a cell membrane gene from a plant by putting in a gene from an animal, I don't think we've got to that point yet. I mean, it's an interesting question.
Yeah, excellent question. Right. Oh, look, there's one more. Hang on. We just got time for 1 or 2 more questions.
That's fine with me.
This is one more about the, are there many modules on this subject or related subjects in the biology degree?
Well, yes and no. So it depends which path you're on. So if you're doing biology or zoology, one of your compulsory modules will be 2nd year evolution, and we will touch on things like domestication, and we'll compare domestication, which is sort of human selection, to natural selection in terms of more sort of Darwinian evolution and things. And then in the 3rd year, I do a module called evolution and genetics, and basically 1/3 of that module is all about domestication, so you will get all this, but then 1/3 of that module is about evolution in the wild. So we do talk about whether the modifications that humans have made to domesticate species can be considered the same as modifications that go on in the wild, making plants or animals adapted to their environments. So definitely in 3rd year you'll get the option to do this, and I think anyone across the school can do evolution and genetics. In the 2nd year, if you're biology, ecology, zoology, you'll definitely do this because it's a compulsory module. And of course, compulsory evolution does make absolute sense for anyone doing
it does.
Well, anyone doing anything, in my view, but certainly biology and ecology and zoology.
Yeah, so the answer is it depends on the program really that you come in, but don't forget that if you come into the school on 1 degree program, you can and there's a lot of flexibility, so you can swap between programs as you go through. I think the other thing might be worth flagging, Mark, is that you do take third year project students who work on projects like this, so you can actually be connected to Mark's research if you choose to do a project with him. So you did a project last year with students looking at, was it aubergine domestication, I think? Or...
Yeah, we've done lots of work on aubergines. We've done work on brassicas. This year, I've got some undergraduates who are doing a project. They're sequencing a genome. So 5 biology, or I think they're all biology undergraduates. They're all sequencing a plant genome. So some of the work I do is on sort of the whole genome scale of things. And we've got 5 plants. They're crops, but they're grown in parts of the world where they end up being very drought tolerant or heat tolerant. But internationally, they're not very well known. And we call these underutilized crops. So there's something called a zombie pea. There's something, there's a sword bean and a jack bean. If you Google them, they're not as exciting as they sound, but they're all these legumes that are incredibly drought or heat tolerant. But internationally, they're not very well known, not compared to normal peas and beans that you might know about. But in my lab, we do genome sequencing. So I've got 5 undergraduates who are going to sequence one of these plants. And we're hoping actually to publish it as a research article as well, because it's all novel data that's important to the field.
Yeah. So the students will be authors on some original scientific literature. And I think that's a really exciting thing, to think that you're contributing to the scientific knowledge base while you're receiving your education. You may not go on to, be a scientist, but even feeling that you've had that opportunity to contribute to the knowledge base in a really important area like that, I think is great. And then just lastly, I think we need to make this the last question. Because I can't see any more in the chat anyway. Are there transgenic topics on the Southampton biology course? Yes, is the answer. So we talk about genetic modification, transgenics, cloning. Students actually get hands-on experience of doing various kind of molecular biology techniques in the lab. So I'm just trying to think of a specific example, because it's spread through quite a few different modules, isn't it? Yeah. But I certainly talk about transgenics when I talk about C. elegans, Sinorhabditis elegans, which is a model genetic organism that I work on in my lab. So it cuts across a number of different research areas in the school.
And we do have third-year projects doing CRISPR as well. So actually using these techniques, I don't know in what organism Nullin is doing this, but I think it's more in cell lines.
It's in cell lines, yeah.
But potentially you'd have an opportunity to do some of these incredibly modern technologies that we're doing as researchers, and we're publishing and we're getting grants, thousands of pounds to do some of this research. And we sort of, we want you to be involved in that and take part, because we are research led. We want to tell you about our research, and some of the stuff I might tell you in one of my modules, it might be something we just did in the lab 2 weeks ago and we just found out. We haven't even had a chance to tell everyone yet, so you'll be the first to hear about it.
Yeah. So thanks, Mark, for that lecture and answering the questions, and thanks to all our guests for coming along. I hope you found that interesting. And hopefully inspirational. Maybe you'll be the people working on food security for the future. Certainly a very important problem, and the world's never needed talented biologists more than it needs them in this century, I think. So it's really lovely to have you with us this evening, and I think we can finish there. So it just remains for me to thank you all very much indeed for coming along, and keep in touch. And if you've got any questions about your offer, then do contact Amrit Mudha, our admissions tutor. Thank you very much for coming. Bye.
Thanks everyone. Bye-bye.